think back to our antibody structure. Antibodies are these Y-shaped molecules. And recall where our hands would be. What part of the antibody do we call this? That contacts and grabs the antigen. It's distinguished from, we have the part that's our legs, and then we have this part. The variable portion. Now, it's kind of odd because you would think this is the antigen binding portion, which it actually is. So why would they call it the variable portion? Versus, why would they call the legs of the antibody the constant fragment? Well, that's because you have only a certain number of genes in your genome that encode for antibodies. And so the question has always puzzled scientists, how can we make so many different antibodies that can recognize almost any antigen that might exist on the planet, but we only have so few genes? How is that possible? And there are going to be two major ways in which we alter the variable region, and then we're going to see a third way that we can change antibodies that's actually going to modify the constant fragment. I know that kind of sounds like an oxymoron, modifying the constant fragment, but you'll see how we do change that as well. So one of the first ways that we build a tremendous amount of variability into the antigen binding domain is by a process that is called somatic hypermutation. Now, once again, this only occurs in the variable part. So somatic refers to the body, and we usually distinguish somatic from a germ or a sex cell. So these aren't your stem cells that are going to give rise to, your stem cells give rise to B cells. But as B cells begin their maturation process, in these somatic cells, this is when the genes are going to start to change. And the way this change happens is when you look at the gene for the variable region, and again, antibodies are protein. We see that the DNA, we're going to have cysteines. And these cysteine residues will lose an amine group. And when you deaminate a cytosine, I was saying cysteine. When you deaminate a cytosine, that converts it into a uracil. Now, where do you normally find uracil inside a cell? Is it in the DNA? No, it's RNA only. So now you basically have a mutation. Now, not all cytosines undergo the deamination. And not the same cytosine in every cell will undergo the deamination. It's completely random. They may all do it. None of them may do it. And some of them may, and it just depends on that particular cell and that particular day. So now as this cell is cloning itself, we have to replicate the DNA. Remember, that's going to be in the S phase of the cell cycle. And so as we replicate the DNA, we've got DNA polymerase. Right? DNA polymerase reads the template strand and it assembles a new strand. So when that DNA polymerase is reading along, it sees, oh, there's a G in the template. What's it going to put as the complementary base? What's the complement to a G? C. All right. Here's a T. What's it going to assemble in the new strand? A. Here's a A. What's it going to assemble? A T. Now, normally, when a DNA polymerase comes on an error, it's able to repair it. DNA polymerases have proofreading activity. But now, this particular DNA polymerase in our cells, in our B cell lineage, it's going to see a uracil. And instead of taking the time to repair that damage, this is kind of like an hourly employee. It's going to go, you know what, I'm not going to call the supervisor and wait for them to come over here to tell me how to fix this. This DNA polymerase just reaches in its bag, gets any nucleotide, puts it down, and just keeps going. So you randomize which cytosines become uracil, and then this particular DNA polymerase randomizes which of the 4 nucleotides is going to go into the new strand. Now do you see how we're shuffling the deck and we're changing the gene at the nucleotide level for that next cell? So in our illustration, we have our variable region. You can see our cytosine, changed it to a uracil. Now we've got an open spot because we had to remove the uracil. But now instead of that C being in place, there's a T, just random one of the 4. So this happens all throughout the lineages of our B cells. And that's how these antibodies, even though they might be similar, they're going to be different. Hence, the term for the variable region. So this is just an example of the cloning process. And so 6 days after exposure to an antigen, you've caused a B cell to begin to clone itself. This B cell had its receptor. The antigen bound to that receptor, so you know that cell can bind to that antigen. And what started out as one single cell, now we have 1, 2, 3, 4, 5, 6. You have 8 different clones of that B cell. And each of these colored little lines, these hashes, marks a mutation. The colors designate one of the 4 nucleotides. So that's after 6 days. We go into days 18 through 16, we have even more mutations. But once we get into this middle range, notice that 3 of our 8 disappear. Now, this is referred to as negative selection. Why do you think 3 of these 8 disappear? Remember, we've got the receptor that can bind antigen. But as we mutate our gene and change the protein, invariably, what are we going to do to that protein? As we're randomly making these mutations for these genes to make antibodies, invariably, we're going to change that region so much it can no longer bind to the antigen. And so these 3 basically are antibody cells, antibody-producing cells that make antibodies that will no longer bind to the antigen we need it to. And so that's why they just kind of go away in our illustration. They're no longer effective. And so when you think about these mutations, it may seem anti-intuitive, because if you have an antibody that already binds antigen, why are you doing the mutations on it? Why would you do that? It already worked. If it's not broken, don't fix it, right? Have you heard that old saying? So how many of you in here have iPhones? Okay, you got an iPhone? You got an iPhone, Samsung? Got an iPhone? Okay, so raise your hands again. Okay, raise them up high. Okay, we've got unanimous iPhone users in here. Okay, no, keep them up. All right, keep your hands up if your iPhone is iPhone 1. If you're using an iPhone 1. Mutations are looking to make it even better. But in the process of trying to improve the antibody, invariably some are not going to work. But look what's happened to 2 of these. Do you see in number 2 here and then second from the bottom? All of a sudden, these things are expanding like crazy because they have a foundational set of mutations that somehow made these even better than the original. And they're expanding, but as they expand, what are they still doing? They're still undergoing the mutation to try to get even better. Now, I'm kind of anthropomorphizing because it's not as if they know, ooh, we're better, we need to do it fast. No, it's just the selection process. They're the ones that are capable of binding to the antigen better, and so they're going to clone up faster. So do you see how the random mutations serve a purpose in, number one, the variability that you can include, but also the efficacy of a particular antibody? How efficient is that particular protein at binding to antigen? So our variable region, those genes undergo somatic hypermutation. That's the first level of variability. The second level of variability we refer to as VDJ recombination. Because in the B cell genome, there is not a single gene for the variable region. Hear what I said? There's not a single gene for the variable region, but there are multiple variable region genes. And the particular cell that's undergoing the replication process, it will choose which variable region to use. And so there's a random selection for which actual block of DNA a particular cell is going to use. So think about gene splicing, introns and extrons. Exons, did I say extron? Introns and exons, this is from intro biology. You keep the good stuff and you splice out the stuff you don't want. Well, that's what we're going to do. We're going to select these cassettes for the variable region. D is sort of an intermediate piece of DNA, and J stands for the joining portion that's going to join this segment to the constant fragment. So first, you select your D and J segments, and then you're going to plug in the V segment, splicing out everything in between. So when we look at our illustration, here we have 4 cassettes for variable, 4 for the D region, and 4 for the J. There's our yellow constant fragment. In each one of these 4 variable region genes, somatic hypermutation is occurring, but it's occurring randomly, and it's going to be different in each 4 of these. Now, as the cell's beginning to replicate itself, it's going to select one of those 4. So do you see the next level of randomness? It's like dealing a deck of cards, but you've mixed in 4 decks of cards, and so you never know which card you're going to get for which deck. That would make Texas Hold'em even more crazy. So variability is built into our immune system because of, 1, somatic hypermutation of the variable region, but number 2, having multiple variable region genes from which you can randomly select. And every cell, every B cell, will do those 2 things. And so as you think about the thousands and millions of B cells you will have in your body, randomly altering these variable regions, it's not a surprise that your body can essentially create an antibody that can recognize any antigen that it might come in contact with. But the trick to that is that you may only have a few B cells that could particularly recognize a particular antigen. So you've got to get that antigen in contact with that B cell to mount an immune response. And sometimes that's the trick. And clinically, we can sort of stack the deck in our favor to have more cells of a particular type, just in case we encounter an antigen in the future. Now, those 2 mechanisms are how we change the variable region. Here, we're looking at a mechanism for changing the constant fragment. So far we've been using different hands, different gloves. What are we going to grab? Now we're sort of putting it on different machines that are going to have different functions, in fact. and confer different properties to these antibodies. And so we refer to this as isotype switching. We're going to see about 5 or 6 different isotypes of antibodies, and they're going to be different gene blocks that we'll plug in. And these isotypes are used at different times during an immune response or for certain locations in the body where you need very, very tough antibodies. So when we look at the antibodies that we are going to remember as far as their isotypes, we're going to have a number that we can run through. However, it's interesting, their expression during an immune response is going to correspond to their position on the chromosome. And so as we look at this illustration, here's our VDJ that we've already spliced together for variable region. Here are our different cassettes for our isotype and the constant fragment. We're going to see that the first two, 5', are going to be the M and then the D isotypes. After M and D, then we get into the G isotype. That's the main one. But notice in this particular illustration on the left-hand side, we've spliced the VDJ. We got rid of the M and the D, and we're going to splice the VDJ to the first G cassette. Here, we're splicing out the M, the Ds, and the Gs. Now we're going to splice it to the fourth, the A cassette. Now, here's the thing I want you to appreciate in this picture, and it's important when we talk about the function of antibodies. We've got this VDJ cassette that we can link to any constant fragment. And the first isotype we're going to use when we're first exposed to a pathogen is going to be the M, because it is the 5' gene on a chromosome. So as we have B cells that are producing plasma cells that make IgM type antibodies, that plasma cell can then later produce any of the other genes because they're downstream. They're 3' on the chromosome. They're still present. But look in this illustration. When we splice out the M and the D so we can begin to produce the G isotype, we can never go backwards. If we're producing the G isotype, we can never again in the life of that cell produce the M and the D because that gene is gone. We got rid of it. Does that make sense? We can go downstream, we can move to the right, but we can never move back to the left. So here in bold are the isotypes and the order in which I want you to remember them. M, D, G, A, and E. Notice we have others that are not bolded that have numbers beside them. Those are subtypes. We're not going to worry about the subtypes. We're just going to worry about the major isotypes. M, D, G, A, and E. And left to right is 5' to 3'. That's how they exist on the chromosome, and that's the order in which you see them produced during an immune response. So let's take these in order and talk about their structure and also their function. An IgM, this is the first antibody to be produced in an immune response. You're actually going to find this on the surface of many of your cells as a B cell receptor. You're going to find IgM in your plasma. Because in fact, when we get to the chapter on blood types, which is our next chapter, we're going to see that principally the antibodies that cause you the biggest problems and the reason you can't just necessarily get blood from anyone are because of the circulating IgMs that recognize either the A factor or the B factor on your red blood cells. And if you didn't have circulating antibodies, then you could essentially get blood from anyone. But it's the antibodies that are the real problem, not necessarily the red blood cells. Now, when we first started talking about antibodies, I stood here like I was doing the YMCA and made the Y symbol. That is our typical picture of an antibody. Well, now take 5 of these and tie their feet together. And what you're going to end up with is a pentamer of antibodies. That pentamer is the IgM structure. It's essentially 5 antibodies tied together at the constant fragment. Therefore, they have 10 antigen binding sites. Because an antibody is bivalent. You got 2 hands. It's not surprising, with IgM being the most 5' on our gene, that the IgM is the principal antibody that's produced the very first time you're exposed to antigen. That's called a primary immune response. So there is our large pentameric IgM. And again, remembering antibodies are bivalent, you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 variable regions that can bind antigen. And it's very possible that you may bind 10 pathogens. You may tie them together because each variable region will bind the antigen on a different pathogenic organism. And so now, instead of handcuffing 2 organisms together, now you've got 10 of them captured and anchored together. Do you see how these can form really huge clumps of material? And that's how your blood can clump up if you are given the wrong type of blood in a transfusion. That will be a bad day if you get the wrong transfusion of blood. So after the IgM isotype, the cells are going to switch and they can begin to produce IgD. This is the second. Now, when we look at the IgD type and we look at the receptors on our plasma membrane of our B lymphocytes, we're going to see that about 1% of those receptors are going to be the IgD type. IgD is thought to be the signal that, okay, I'm ready to recognize antigen now. And unlike IgM that is a pentamer, IgD is that typical monomeric Y-shaped molecule on the surface of our plasma cells. So with an IgD, that's almost like the open close sign on businesses. Those IgDs mean I'm open for business. Come on, antigens. I'm ready to be activated. I am immunocompetent. I'm ready to begin to produce more cells, memory cells, plasma cells, and fight off the infection. So a different sort of illustration of a monomeric antibody. Again, you can see the loop nature of the protein here and the red lines being our disulfide bonds. You can see how we tie the light chains in blue and the heavy chains in green all together into that Y-shaped molecule. Now, after we go through M, we get to D. Our third isotype is the G. This is where we get the immunoglobulin. And the IgG type, beyond being a monomeric immunoglobulin like we saw with D, this is the most abundant immunoglobulin in the serum of your blood. In fact, 75% of those proteins are the IgE isotype of antibody. And while IgM is the first to appear in your serum when you first encounter an antigen, let's say that chickenpox virus, the first time if you had it when you were a kid, well, later in life when the chickenpox virus comes around again, the predominant antibody that's produced and found in your serum is not the IgM, but the IgG. It's the most prominent in a secondary immune response. Now, IgG is pretty important as well because the IgG is the only isotype that's produced in the human body that can cross the placenta. And so when a baby is born, the baby has passive immunity. Passive in the sense it got antibodies from mom. Whatever mom had antibodies against, the baby's going to be born with those antibodies. But once those proteins get broken down and eliminated from the body, that baby doesn't have the B cells and the plasma cells to make new ones. So do you see how that's the passive immunity? The baby got the antibodies ready-made from mom, and that crossed the placenta. Again, some other views, a space-filling model on the left of an IgG, monomeric antibody, and then another showing the disulfide bond chains within the chain, and the green showing between the chains and how they're tied together. Now, babies can not only get antibodies through the placenta from mom, but babies can also receive passive immunity through breast milk. But the antibodies are slightly different. The variable regions may be the same, but the constant fragment of antibodies found in breast milk is the IgA type. And IgA is a dimeric antibody, 2 antibodies tied together at the constant fragments. So you have 4 binding sites. These antibodies are much more resistant to degradation because of the different enzymes they're going to encounter in the mucous membranes of your mouth where your saliva is present, in your stomach where you have hydrochloric acid and enzymes. And so as the baby nurses, the very first secretions from the breast of the mother are not going to have a lot of milk proteins in it. In fact, it's fairly translucent. That's going to be colostrum or first milk or immune milk. And that's going to be the secretion that's largely rich in the IgA. Now, is that a problem for the baby? The fact that it's not getting a lot of protein right away? Because when babies are first born, the first couple of days, what's kind of interesting about babies? They lose weight. They pack on about 1 pound a week in the last couple of weeks of pregnancy. They store a lot of fat, principally brown fat, that's going to be very thermogenic. It gives them a lot of their body heat. They burn through that fairly quickly, and it's not long then before the breast starts producing the milk proteins. They get the milk, and then they don't stop eating until they're about 40 years old. But that's the first milk, and you can see an IgA is just the 2 monomers tied together at the constant fragments. And it's possible that that orientation helps them to resist degradation that may occur possibly at the point of the constant fragment. So we've gone through 4 isotypes. The 5th and the last is the IgE. Monomeric, like D, like G. And we see that the IgE isotype, it basically elicits the most powerful of all our immune responses, which begs the question, if this gives us the biggest immune response, why isn't it the one most widely expressed in the body? Why is it G and not E? Well, it's a good thing it's not, because these are the ones that sometimes inadvertently get stuck on the surface of our mast cells. And when we have antigens that bind to these IgE antibodies stuck on the surface of our mast cells, it can cross-link. It can pull 2 of these antibodies close enough together that these IgE receptors in the membrane of our basophils become activated. And when they become activated, they massively release all of the material in their granules, including histamine and heparin. And it's in this case you can have different levels of type 1 hypersensitivity, aka an allergy. If you have a ton of these mast cells that release systemically your histamine and heparin throughout your body, at one time in a huge bolus, that can actually even be life-threatening. So yeah, that level of an immune response, it's a good thing it doesn't happen all the time. It would make for an extremely bad day. So here we see an example. Here's our antigen presenting cell. See the IgEs and allergens, but here's where we have a problem. This is how it's supposed to work in our antigen presenting cell, but when we get these linked on our mast cells, we see the massive release of cytokines, prostaglandins, leukotrienes, histamines, heparin's also going to be released. When that single allergen can pull those two antibodies close enough together and those receptors can activate each other, it causes the degranulation of the basophil. Because think about heparin flooding through your body. It's going to be an anticoagulant. Histamine is going to be a vasodilator. The prostaglandins, leukotrienes, they're going to increase vascular permeability. So your blood vessels are going to open wide open. Your blood vessels are all going to start getting leaky. And what's going to happen to your blood pressure? It's going to plummet. You're going to get inflammation because of all the leaking fluid. If that inflammation occurs around your airway, what are you going to have trouble doing? Breathing. So I think you can see that type of allergic response, it's not a good thing. It can be very, very dangerous. So that's how we make antibodies so variable. That's how we can change the location and function where we see antibodies located. Now let's look a little bit more closely at what antibodies can do. We already know opsonization. This is where antibodies decorate that pathogen. Opsonization means it makes it yummy for phagocytic cells. You remember my word, chocolatification? When you coat a pathogen with antibodies, macrophages cannot resist eating that material. And so, yeah, antibodies, those FC fragments that are sticking out, irresistible to macrophages. Completely attracted to that FC portion. So yeah, phagocytosis of our granulocytes and especially our macrophages is absolutely enhanced when you slap all those antibodies on the surface of a particular pathogen. But what can also happen when you have all these antibodies decorating the surface is you can link certain proteins together in such a way that you create an activation factor of proteins, and this activates another set of plasma proteins that are in an inactivated state. And these are called your complement proteins. And this complement cascade is going to look like that domino series cascade we saw with clotting. But in this case, we are going to begin to cleave these proteins, activate proteins in a sequence. And so some of our activated complement proteins are going to be able to enhance opsonization. They're going to enhance the phagocytic activity. They're going to increase inflammation. And some of these complement proteins are actually going to be able to form pores that can insert themselves into the plasma membranes of pathogens and destroy them lytically. Just essentially poke holes all in the pathogen until they're dead. So antibodies opsonize, they activate complement, but we also see that antibodies can actually cause cellular toxicity. And so sometimes antibodies on the surface can cause the pathogen itself to release products that attract our granulocytes that then produce substances like superoxide ions that can cause that pathogen to be destroyed without actually making physical contact. So again, like throwing hand grenades, especially our killer T cells, our cytotoxic T cells. We're going to talk about those after we finish all of our antibody type work. So here we can see our antibodies. We can see they decorated this infected cell. There's our natural killer cell that causes the lysis because the antibodies were recognized. Here's our complement activation that comes over and just simply inserts itself and destroys the cell. There's the increased opsonization. We're going to put this complement all together in a flow chart and a cascade that we'll see. But the things I want you to remember: antibodies, opsonization, complement activation, and antibody-dependent cytotoxicity. Opsonization we got. Complement activation. We're going to see in our cascade that we've got these complement proteins in the serum, C1 through C9, held in an inactivated state. And sometimes when antibodies decorate the pathogen, that can lead to the activation of C1. C1 is going to form a complex with FC fragments and other proteins to become activated. C1 then is an enzyme that can go on to cleave and activate C4. That cleaves and activates C2. That you see the domino effect again, like we had the clotting cascade. C2 is going to activate C3. C3 in many ways is going to be like factor X. You know that point in which these two pathways converge? C3 is going to be a convergent protein again for two pathways that we're going to talk about. And C3 fragments are going to be able to help opsonize. They're going to be able to help with inflammation, among some of these other fragments. But C3 is also going to go on to cleave C5 through 9, and it's going to be these complement 5 through 9 that are going to form the pores, which we call the attack complex, that are going to insert themselves into the membranes of pathogens. What we've talked about in that particular slide is called the classical pathway. You have the antibodies that have opsonized the pathogen to activate C1, C4, C2, and there's our hinge point, C3. Now, the nomenclature is such that when you cleave and activate, you basically cut these into two parts, an A part and a B part. So you see in the lower left, C4A, C3A, and C5A, the A fragments of these complement proteins, are going to promote and enhance the inflammatory response. It causes blood vessels to become more permeable. It causes vasodilation. It causes our mast cells to release more histamine, and so it's like a positive feedback mechanism. But once we activate C3, the C3b portion, you can see, comes over and it enhances opsonization. Well, we've already opsonized with antibodies, right? That's like the chocolatification, make it yummy. Well, the C3b is the whipped cream. And you know they make chocolate whipped cream? Yeah. So you put the milk chocolate on, then you put the chocolate whipped cream on. You better get out of my way if I ever see that dessert. And that's what happens with our phagocytic cells when they encounter these opsonized and enhance extreme opsonization, I guess you could say now, with our C3b fragment. So enhanced inflammation, enhanced opsonization, but C3b also activates 5 through 9 that forms a pore complex called the attack complex. It's going to look like a little barrel of proteins that inserts itself through the plasma membrane of the pathogen, causing the cytoplasm to leak out. So cytolysis, the cell just bleeds to death in effect. And all of that is because we began the process because antibodies had opsonized our pathogen. So while classical pathway starts because of antibody opsonization, we have that right-hand pathway. It's referred to as the alternate pathway. And it's going to begin when you have unique carbohydrates. Some would call them xenobiotics. foreign materials. So some bacteria have unique carbohydrates that our body doesn't recognize and our body doesn't produce. And when these are encountered as part of our immune system, that leads to another cascade, C3, B, and D. It's a much faster process. But again, the end result is the same, splitting 3 into A and B fragments, going on to enhance inflammation, enhance opsonization, and form the attack complex. I like this illustration because when it breaks them apart, it looks like a little capsule. You know, your capsules have the 2 halves. And especially here at the bottom, you can see how 5 through 9 form this little barrel that's inserting itself and causing the cell to hemorrhage its cytoplasmic materials. And when that happens, the cell dies. That's our cytolysis that happens because of the attack complex. Now again, a lot of information in one slide. If you notice, we've got the classical pathway on the left, alternate pathway, those xenobiotics, the carbohydrates on bacteria. And in the middle, there's a third pathway. Don't worry about the middle. All right, lecithin is going to be another type of carbohydrate, but we're going to stick with the classical and the alternate. C3 and what C3 does, the A fragments, the B fragments, and then our attack complex on the far right.